Happy morning to one and all present here on this online forum. I, on behalf of KTN College of Pharmacy, cordially welcome all to the 7th International Webinar entitled Pharmaceutical Sciences in Tissue Engineering and Regenerative Medicine by Ramesh, USA. Ramesh, I think in the year 2004, I took a class on pharmaceutical technology, if I'm correct, and you are in third year B pharmacy at the time. I thought that this seed will grow into banyan tree. Of course, you all could see the banyan tree on the other side of the screen. And he is such a well-mannered, matured, motivated, humble, honest, hardworking candidate with a proven academic and research record. That is very, very important. I wish you all the very best, Mr. Ramesh. Stay home, stay safe. Over to Jesh. Thank you.
Dr. Ramesh, born to the proud parents of Mr. Subbiah and Mrs. Chandra in the historic city of Tenkashi. Dr. Ramesh graduated in pharmaceutical sciences from the Tamil Nadu Dr. NGR Medical University, Chennai in the year 2005. That took Dr. Ramesh much ahead as an MS candidate in the Department of Bio-Nanotechnology, Gakon University, South Korea in the year 2009. After completing his MS program, Dr. Ramesh served as a visiting scientist for a year in 2011 in the Center for Nanomaterials, Korea Institute of Science and Technology, Seoul, South Korea. In the year 2012, Dr. Ramesh advanced in the same institute at the Center for Biomaterials, but this time as a doctoral candidate for biomedical engineering. And this doctoral milestone was achieved in 2006. After the doctoral program, Dr. Ramesh persuaded for his postdoctoral fellowship, not one, but three. First postdoctoral fellowship was secured from Korea Institute of Science and Technology and Chunghang University, Seoul, South Korea in the year 2017. The second from GWW School of Mechanical Engineering, Georgia Institute of Technology, Georgia in the year 2019. And presently he is in the final stages of his third PDF from Oregon Health and Science University, Portland, USA. Furthermore, adding to his credits, he has in his name 36 peer-reviewed publications, 4 peer-reviewed review articles, and he has authored 4 book chapters, and above all, a patent in his name. Dr. Ramesh fetched various research grants from premier organizations, some of which includes the Armed Forces Institute, Ministry of Science and Technology, and so on. He's also a recipient of prestigious awards and honors right from the beginning of his career. He has been an invited presenter in many conferences around the world. Meanwhile, he has presented his research works in a humpty number of oral and poster presentations at conferences held in USA, South Korea, and other parts of the world. He is a mentor for undergraduate, master's, doctoral trainees of different international universities. He is a teacher and above all, a leader with vast experience. He is also a member of various professional societies. And last but not the least, the essence of research. That is, he is a reviewer as well as an editorial member of high impact factor journals. With this little introduction, I now invite our dynamic speaker, Dr. Ramesh Subbiah, to take over the session. Sir, over to you, sir.
So today we would like to discuss about how pharmaceutical science can benefit tissue engineering and regenerative medicine. So the field tissue engineering and regenerative medicine is pretty new, and it includes like multiple departments, basically helps grow this field forward. One of the field is pharmaceutical science, which contributes a lot to advanced tissue engineering and regenerative medicine field. So we will discuss about what is tissue engineering, what are their components, what are the advancements currently happening behind this field. So to begin with, human body. So human body consists of billions of cells that collectively build functional organs with systematic physiological functions. Active function of organs depends on cells by consuming nutrients and convert them into energy. Every cell types are unique in exhibiting their own functions, and cells undergo several mechanisms to self-regulate and to be functionally active. What happens if there is a malfunction of cells? Malfunction of cells leads to organ failure and cause of death if untreated. The number of organ failure is in an increasing order every year. Besides, chronic diseases including congestive heart failure, liver cirrhosis, renal failure, and then osteoporosis, diabetic mellitus, all these factors cause harmful to life. Therefore, reconstructive surgery have emerged to save life by transplantation using autograft or allograft. About 666,000 transplants have been carried out during the period of 1988 to 2016. This data is from organ procurement and transplantation networks. Severe trauma is hard to be treated because of the size of the defect. Although bone has endogenous capacity to heal itself, large defect and severe trauma, ischemia and infection requires augmentation of treatment. When we say augmentation of treatment, we use autograft, which means we take part of our own body for the replacement of the defect site with the addition of vascular flap or growth factors such as bone morphogenetic protein 2. The limitation of this treatment strategy is we don't have enough graft materials available to heal the defect site. And also the outcome, regenerative outcome is very, very poor. In addition, extremity trauma includes 50 to 88% of combat injuries, war injuries. And then 1 in 7 of complex injuries result in delayed amputation. That means we have to remove the limb, the affected part of the body, which is associated with high morbidity, revision surgeries, and so emerged to treat or to regenerate or to repair such severe trauma cases. So that field called as tissue engineering. This field is rapidly emerging. This is a rapidly emerging biomedical research field. This provides an alternative to grafting and transplantation and reserves true potential to confront the transplantation crisis caused by the shortage of donor tissues and organs, and also to meet patient needs and improve patients' lives. Since last decade, the field of tissue engineering and regenerative medicine has received much attention for the proven effects on tissue replacement, repair, and regeneration using combination of multiple factors. The foremost important one is stem cells, and then growth factors, and then biomaterials. Advanced drug delivery system for biosignaling molecules, in vitro cell manipulation, and engineering biological functions of cells, and 3D printing are the key strategies applied in tissue engineering currently. So these are, whatever we are discussing, these are like high-tech, cutting-edge technologies. And we will discuss some of the core technologies that has been used for tissue engineering and regeneration today in this webinar. As I said before, cells are at the forefront in regeneration field, particularly stem cells, which are a class of immature and undifferentiated precursor cells that are able to self-renew and differentiate into specialized cell types. The source of stem cells are embryos formed during the blastocyst phase of embryological development, such as embryonic stem cells. And also, adult tissue has a lot of stem cells. They are called as adult stem cells. And other type of stem cells also available. Although embryonic stem cells are totipotential, that means embryonic stem cells can become literally into any type of cells. But experimental using these cells are prohibited in genicity and rejection. In contrast, we have adult stem cells such as hematopoietic cells and mesenchymal stem cells. They are found in mature tissue of our body, such as bone marrow, and then placenta, umbilical cord. Those kind of tissue has enormous amount of stem cells. They are called as adult stem cells. They are capable of generating multiple lineage cells and can be used for organ regeneration. As we see here in the chat, adult stem cells and embryonic stem cells can generate lung cells, pancreas cells, blood cells in the lab. Recent clinical studies have demonstrated the feasibility of stem cell therapy in tissue repair. But there are many unresolved questions still open for future tissue engineering research, particularly controlling or manipulating the extracellular matrix microenvironment that determine the stem cell differentiation process. So what are extracellular micro environments? We will discuss about that now. So stem cells, as I said before, they can differentiate, but they cannot differentiate to function cells by its own. They need additional factors. When you say additional factors, we have chemical stimuli, physical stimuli, and then extracellular matrix, cell-cell interaction. So stem cells interacts with numerous cues or factors for self-regulation and differentiation into target cells. This image shown here shows these are the possible microenvironmental factors can determine stem cell differentiation. For an example, when the stiffness of the material is high, stem cells tend to differentiate into bone because bone is a stiff material. When it comes to fat cells, fat is a very soft muscle. So if the material we culture on if the material is soft on which we culture mesenchymal stem cells, that will become fat cells. So this is how these microenvironmental factors play a role in differentiating stem cells. One of the important differentiation agent is growth factors, which is a biochemical stimuli, which is called as biochemical growth factors. So what are growth factors? So growth factors are nothing but cell signaling molecules. There are 4 categories of growth factors secretion found in multicellular organ signaling and direct signaling. The main difference between the different categories of signaling is the distance that the signal, that molecule, secreted molecule, travels through the organism to reach the target cells. Not all cells are affected by the same growth factors. So now talk about growth factors. They are soluble proteins secreted by particular type of cells that actively participate in cell signaling pathways and regulate cell behavior. A number of growth factors have been recognized along with their specific transmembrane receptors. First, growth factor beta superfamily, which consists of nearly 33 members. As all these factors are secreted by a particular type of cells to repair and regenerate naturally in our body. And during the developmental process from fetus to a baby growth, all these growth factors play an important role. So understanding growth factors more, we can utilize them for the regeneration of tissue. So the bigger question here is, can growth factors regenerate tissue? And then to achieve successful regeneration of growth factors, next generation delivery systems are needed. Can they effectively deliver growth factors? Then the third question here, why vascularization is important in tissue regeneration. So when we regenerate tissue, for an example, bone will regenerate, but without vascularization, the regenerated bone will die. So vascularization is very important. So these 3 questions we are going to address here. And then functions of growth factors. As I said before, growth factors play a critical role during development. So meaning from the embryo formation to the fetal development, multiple growth factors are secreted and they have self-renewal, migration, and differentiation. Growth factors bind to a cascade of intracellular signaling events through transmembrane receptors that culminate in both transcription-independent and transcription-dependent changes in target cell behavior. Number of studies have been tested and they demonstrate that individual growth factor has positive effects on the differentiation of stem cells, such as insulin-like growth factor on beta islet cells, beta for the secretion of insulin, BMP4 on lung progenitor cells, BMP2 and BMP7 on osteogenic cells, which are bone-forming cells, VEGF, vascular endothelial growth factor on endothelial cells and smooth muscle cells that forms blood vessels, and then TGF beta 1 and TGF beta 3 on chondrogenic cells, which are basically cartilage. So if we have a defect in our body, cartilage repair, cartilage injury, bone injury, or blood vessel injury, we can deliver this particular protein to regenerate or to repair. Very sensitive. There are multiple factors affects the regeneration effect of growth factors. So growth factor can be, so we need to have a fundamental understanding about the properties of the factors affecting delivery system from the perspective of growth factors, biomaterials, and carriers to realize the potential tissue engineering application of growth factors. For growth factors, dose optimization is critical factor to maintain the therapeutic dose at the injury site throughout the period of healing. For example, if there is a severe bone injury, it takes 2 to 3 months to heal. So if we implant something, a material with the delivery system, that should be able to achieve growth factor delivery more than a month in order to achieve successful regeneration of the bone. And when it comes to biomaterials that's been used for the implantation, solubility, cytotoxicity, surface charge, and rate of the system, what is the size, loading efficiency, and what is the release pattern? These properties are important. And then biomaterials are generally used for the fabrication of ideal delivery system to deliver growth factors. So what are biomaterials? As per Society for Biomaterials, they explain biomaterial is a material intended to interface with biological system to evaluate or treat, augment, or replace any tissue, organ, or function of the body. Biomaterial or synthetic biomaterial, which are further divided into cationic natural biomaterial, anionic natural biomaterial. Likewise, synthetic cationic biomaterial, synthetic anionic biomaterial. These are the widely recognized biomaterial used as a delivery system in tissue engineering and regenerative medicine. So this is very important because naturally, if a cationic molecule will naturally attract the anionic molecule. So if we choose natural biomaterial, which is cationic, in case of keratin, we can use this keratin to deliver anionic growth factor so that we can achieve slow, steady, controlled, sustained release over a period of time, which we will discuss in the coming slides. So using these biomaterials, we can prepare delivery system. So what we see here is considered standard delivery system. The selection of the rate controlling delivery system should be simple to fabricate with high loading efficiency, cost effectiveness, and stability for in vivo applications. This delivery system should also enhance bioavailability, minimize immune responses, and overcome pre-systemic metabolism, clearance, non-specific cellular uptake, and secondary damage to cells. There are several properties of these delivery system are important to achieve increased loading efficiency and optimal growth factor release kinetics. That means if we want to deliver 5 microgram of protein for 28 days, so the material that we develop should be able to retain or should be able to release this 5 microgram over a period of 28 days. That is very important. So such as particle, capsule, fiber, or tubes, something like that. And then by their size, are they nano range, nanoscale particle, or micro scale particle, or macro scale particle. And then based on the surface property, such as are they porous or non-porous. Why this is important? Based on the size, shape, and then surface, you can tune the release property of growth factor. As you see here, it can be burst release, it can be sustained release, it can, using external stimuli, you can achieve pulse-like release, and then using cross-linking, you can achieve delayed release. So this is the important function of size, shape, and surface property. Advancement in course layer by layer coated capsules, nanoparticles in fiber scaffold or nanoparticles in hydrogel scaffold, and then scaffold containing extracellular matrices. By utilizing advanced delivery system, you can deliver multiple growth factors in a programmed manner at the same time. For an example, in case of corsal microcapsules or particles in capsules, you can deliver two different therapeutics at the same time, but with different kinetics. And then with corsal microcapsules, you can achieve spatiotemporal release. That means I want one protein to be released early in the first week, and then the other protein to be released sustained over a period of 28 days. And then layer by layer coating, you can achieve pulsatile-like release, like, okay, on, release, off, no release, on, release, off, no release, something like that. And then sequential delivery using, again, corsal structure can be used to achieve sequential delivery. One protein in the first week and the next protein after the first week. Now let's discuss about some of the examples of delivery system fabrication. First, in this category, we have bulk gel or sponge-like materials. Collagen is well known for the fabrication of bulk gel and sponge-like materials. Due to the presence of teleopeptide in collagen, they can easily form fiber-like structure. They will undergo cross-link in the neutral pH and at room temperature. Fabrication of collagen gel that can be entrapped with growth factor or cells. That's why collagen is the widely used biomaterial in tissue engineering. And also, collagen sponge can be prepared using lyophilization, which is basically freeze drying method. And growth factor loading is high with this material, and then then in this category, we have emulsification technique. As you all know, it's a widely used technique and a very popular pharmaceutical technique. Emulsification has been used to prepare nanoparticles to microcapsules of natural and synthetic polymer. What we see here, it's a PLGA nanoparticle synthesized from using emulsification technique. The one on the top we see here is nanoparticles of hyaluronic acid, and then microparticles of methacrylated gelma. In particular, methacrylamide functionalized heparin microcapsule protect growth factor from premature degradation and also minimize burst release and facilitate sustained spatiotemporal delivery. And then we will talk some of the advanced technology. Microfluidic devices, electrodropping or electrospinning techniques, mold-assisted cross-linking, and lithography processes are considered as advanced fabrication systems. Electrodropping method is used to fabricate microcapsule of alginate, wherein applied voltage and a flow rate will play a critical role. So alginate is a beautiful biomaterial that can undergo ionic cross-linking upon comes in contact with divalent cations such as calcium. So alginate consists of two components. One is manuronic acid, another one is gluronic acid. So what happens when calcium comes in? Gluronic acid forms the egg box structure, that is the cross-linking one that forms like a micro gel-like material. That's the beauty of alginate in electrodropping system. And then with an addition of coaxial needle setup, that means we discussed before, can deliver multiple growth factors at the same time. So if you look here, the inner core, the dark color one is PLG nanoparticle that is loaded with one particular growth factor, and the outer layer, which is like a shell, which is made of alginate that can be loaded with a different type of protein. So two different protein can be delivered through this technique. And then electrospinning technique is used to prepare unique fibrous scaffold structures such as nano to microfiber scaffold and particle containing mesh scaffold. These improvements are the direct result of a reduction in the contact time between the biomolecules and the chemicals used for fabrication. Next in this section, we have microfluidic devices to fabricate microgels. Schematic describes the fabrication of microfluidic device with flow controllable geometry, wherein the generated aqueous polymeric droplets is sealed by the oil phase which is then cross-linked by cross-linking agent and form the monodisperse hydrogen. As we see here, through these two outlets, the middle channel, we introduce aqueous polymeric solution with growth factors incorporated. And then with the help of laminar flow, it generates droplets, and then the droplets are surrounded by oil layer, which is then formed like the microcapsule, which can be cross-linked. So number of like thousands of microgels can be produced using these small devices. The size of this channel will be in 100 micrometer. 100 micrometer is the diameter of our hair size. So that's for the comparison I'm telling. So this is the size of the the channel size. And then we have in the pore scaffolds are interesting because they offer 3D structure for cells and growth factor loading. Pore system can be prepared using porogen assisted freeze drying or salt leaching technique, schematically shown here in this slide. And then there are different loading strategies available to load growth factors in the delivery system. What we have seen before, the type of delivery system and the type of fabrication technique to fabricate a delivery system. Now we talk about how to load growth factors. So as I said, there are different loading strategies available. So the first one is direct loading. Then we have non-covalent binding and covalent binding. In direct loading, it's basically absorption or absorption based technique. And growth factor and biomaterial affinities are established by electrostatic interactions, hydrogen bonding, donor-acceptor interaction, and van der Waals forces. And then physical absorption and absorption are two of the three direct loading methods. In both cases, the delivery system is immersed or treated with growth factor solution. Non-covalent binding using ionic complexation is performed by functionalization, and also covalent binding is achieved using linkers and environmental sensitive agent, as well as chemical coupling reactions. So there are a number of loading strategies available to load growth factors into the fabrication system. And then once loaded, how these growth factors are released from the delivery system. So release kinetics can be classified as burst release, sustained release, and then pulsatile release, and then on-site release and programmed release. And also multiple external stimuli can be used to control the release kinetics. In case of physical stimuli, we have sonophoresis, magnetic field or mechanical forces, osmotic pressures can be used to externally control the release of the therapeutics. Some biomaterials are biomaterial exposes to those enzymes, they dissolve and then they release the protein. Then we have environmental, in which case temperature, light, or hypoxia. And then this is the important chart, very close to my heart. So if you look, if we have number of proteins, like number of proteins and the number of biomaterials. So what biomaterial we have to choose to fabricate growth factors. So this chart can be used, it's like a reference chart. So we have anionic polymer can be chosen, one of them can be chosen to choose cationic growth factor, and then we can design a standard delivery system or advanced delivery system to achieve the release kinetics. Within the biomaterial itself, we have multiple parameters that controls, right? For example, some material degrades faster. Degradation faster means release faster. So we need to identify the right material that depends on their degradation and then their surface charge and also their toxicity. So based on this chart, we can identify the right biomaterial, right growth factor, right delivery system for the delivery of growth factor to regenerate tissue. When we think about tissue regeneration, we should have a profound understanding about developmental biology to successfully regenerate tissue. For instance, bone develops from fetal hyaline cartilage model. Cartilage then calcifies and a periosteal bone collar forms around the epiphysis, which further leads to primary ossification center in epiphysis. During this particular process, blood vessels invades, and then the forming bone replaces cartilage and epiphyseal plate to develop into functional bone. A variety of growth factors are involved during this developmental process. This is how from embryo, the bone forms towards the baby delivery. So this process is called endochondral ossification. If you look, number of growth factors participates here, particularly for the calcification here, BMP2, bone morphogenetic protein, plays a critical role. And for the blood vessel invasion, the proteins like vascular endothelial growth factor and platelet-derived growth factor, VEGF and PDGF, play a critical role. So to achieve a functional bone regeneration, as it happens in the developmental process of our body, we developed alginate and collagen-based delivery system. Alginate is a popular carbohydrate polymer, and collagen is a ECM protein, which is abundant in our body. Both are biocompatible, and the delivery system preparation is easy, and they demonstrate excellent loading efficiency, more than 85%, and also demonstrate controlled delivery of growth factor over a period of 28 days. And VEGF, as I said before, VEGF and PDGF are potent angiogenic growth factor. Combination of these two growth factors significantly promoted the vascular network formation, meaning blood vessel formation within the hydrogel. This has been proven by the quantitative data. So we identified VEGF and PDGF at the concentration, at the ratio of 1 to 1.5 achieves, 1 to 1.5 ratio and 1 to 3 ratio achieves the effective blood vessel formation. Then when we deliver dual angiogenic growth factor, when we deliver VEGF and PDGF with low dose BMP2, because BMP2 at high dose causes side effects such as cancer, and then hypertrophic ossification and also inflammation. So we need to use low dose BMP2. So with the combination of dual angiogenic growth factor with low dose BMP2, our system significantly improved the regeneration of vascularized bone tissue. It is very important. Without any of this growth factor, the bone here, what we see here, there will be no bone. That is the empty control, which I don't show here. So only growth factors, single and triple growth factor, effectively promoted regeneration. Particularly triple growth factor achieved significantly improved bone regeneration, and also the mechanics of the bone. We used electrospun nanofiber mesh scaffold, like a cylindrical shaped mesh scaffold in this study to retain the growth factor at the defect site. So result signifies the vital role of multiple growth factor combination therapy on vascularized bone tissue regeneration. This is the recently published article. We published this article in Octa Biomateria this year. Then the same study, the same samples, when we tested histological analysis, such as hematoxylin and eosin staining, trichrome staining, and safranin staining, and scanning electron images, confirmed the formation of mature bone with blood vessels, and then bone marrow, and also mature bone. So results again clearly confirm, right? The combination of growth factors can regenerate functional bone tissue. However, these studies are conducted in small animals. In our case, we use rat, but future studies should be planned using large animals before moving forward with clinical translation. But the potential of growth factor is very promising. Although we could achieve effective bone tissue regeneration in preclinical models such as rats, to date, the biggest challenge in growth factor delivery is control over spatiotemporal release. So what does it mean, control over spatiotemporal release? Meaning, even with the growth factor release, first 1 week I want growth factor A, next week I want growth factor B to be released, which is very, very hard and super challenging. But 3D printing can overcome this challenge. So what is 3D printing? 3D printing is an advanced technique that can be, that has been like recently popularly accepted by the research community, which we will discuss in detail in the coming slides. So here in this one, we have used DLP, digital light processing based 3D printing to fabricate microgels of different shapes like square, flower, and then circle, and then like a spiral type of shape, and also in different shapes and sizes that can be preloaded with different growth factors. And thousands of microcapsules can be prepared in a minute. The hydrogel intended to be prepared in 3D in this particular DLP-based 3D printing should be light sensitive. In this case, methacryl group is being used, which is a light sensitive agent that is functionalized in gelatin. That it's called gelma. Methacrylated gelatin was used for the fabrication of microgel. Also, we have recently developed miniaturized Lego-like micro cages that can be assembled, adjoined, stacked up to suit the complexity of tissue defect in a site-specific manner. For an example, if we have a bone tissue injury in the femur bone, you can get these pieces and like Lego-like micro cages, you can build the defined structure, the defined desired structure to fit them to match the defect size. And very importantly, these micro cages has cargo-like rooms. As you see, these are empty cavities, and these empty cavities can be loaded with growth factors loaded microgels of any type. So you can choose the bottommost layer will carry type A protein, the middle layer will carry type 3 protein, something like that. By doing that, this micro cages also has a lot of porous geometry specific porous that will enhance blood vessel invasion into the core of the defect. As you see here, Lego scaffold with VGF and BMP loaded microgels here in the middle of the core. So greater cell invasion and vascularization. So this is microgel loaded and this is bulk gel loaded. So overall, these studies prove the important role of delivery system, architecture, growth factors in regenerating functional tissue. So we will now move on to discuss about bioprinting. What are the other advantages of bioprinting? What are the challenges with bioprinting and all? So 3D bioprinting, it's an additive manufacturing technique where cells and the biomaterial, which are called biomaterial like bioinks, are deposited layer by layer in three-dimensional geometry in a sterile environment. The greatest importance of bioprinting lies in the resulting tissue-like structure that mimic the actual micro and macro environment of human tissue and organ. This is critical in drug testing and clinical trials, with the potential to drastically reduce the need for animal trials, which is very, very important. When living tissues and organs need not come from humans, this budding technology offers other massive opportunities. One example is testing treatment for diseases using artificially affected tissues. The process could also eradicate the headache associated with organ donation and transplantation. Apart from the lack of available donors, organs, the entire process is criticized, from a more moral and ethical perspective as well. Organ replacement is the main objective of 3D bioprinting, but tissue repair is also possible in the meantime. With BioInk, it's much easier to solve problems on a patient-specific level, promoting simpler operations. So why there is so much excitement around 3D printing? Many are interested in 3D printing research. 3D printing can fabricate any geometric structure, unlike the fabrication technique. Then complex architecture can be achieved. Everything is done with an automated robotic precision, therefore error is minimal. It is easy to use with little skills and truly scalable, meaning it can print from a single cell to a large building. There is not any technology available that offers so much like 3D printing thus far. So we need to understand about the difference. There's a sharp difference between 3D printing and bioprinting. The definition for 3D printing is layer by layer positioning of biological materials, biochemicals, and living cells with precise spatial control of the placement of these functional components to fabricate 3D structure. As soon as whenever there is the involvement of living cells, then it is called bioprinting. Therefore, bioinks are very important. Bioinks are nothing but the ink that is used for printing. So in this case, hydrogel or any kind of biomaterial that will carry cells are considered bioink. are considered bioink. So choosing the ideal bioink is very important. So how does bioprinting work? Essentially, we start with 3D scanning of patients such as MRI or micro CT that will create DICOM file. This DICOM file will be reconstructed digitally. The missing part of the organ will be designed using programs such as CAD. This will be transformed into G code using numerical slicing program. After that, we will choose the ideal biomaterial, in this case bioink, and ideal cells, and send it to the printer for printing. Bioinks are biocompatible hydrogels, as I said before, such as collagen, hyaluronic acid, alginate, and gelatin. Hydrogels are very important component to achieve successful 3D bioprinting. And then let's see a few examples of 3D printing methods. The first and most popular method that everybody knows about is extrusion bioprinting, wherein hydrogel is mixed with cells and loaded in a syringe and pushed through a nozzle to form 3D structure. As we see in the video, the cell mixed hydrogel is loaded in the syringe, which is extruded by the piston to 3D print the design structure. That means the pushing force is high that cause cell damage, and then very limited range of printable inks, that is bioinks, available to use in extrusion printing. Hydrogel with high viscosity and very low viscosity will be hard to be extruded. So these are the challenges or limitation of this method. Next in this category, we have digital light processing based 3D printing. The one that I showed to prepare microgels earlier. So this work is completely different from extrusion printing. It uses light to cross-link anything photo cross-linkable. That means UV light can cross-link particular type of polymers. The most popular photo cross-linkable materials are acrylated or methacrylated or diacrylated. So in this case, methacrylated gelatin is a popular photoresponsible polymer. So this photo cross-linkable material is loaded in the vat or container, and that will be exposed to light, in this case UV light or blue light, through the projector. However, the inbuilt mirror here directs the light path in an on or off mode in a site-specific manner that acts as a mask. That means only particular design. So as we see here in the photo mask, only particular light will be passed through based on the mask. As a consequence, photo responsible polymer is cross-linked based on the mask exposure. So this will be built layer by layer. So in this case, in the video, what we see here is the 3D printing of leaflet. This has its this shows like a beautiful 3D leaflet formation, 3D printed, and which is functional as well. So most of our vein in the leg a particular DLP based 3D printing has its own limitation, such as only photo cross-linkable polymers can be used, which are very less in numbers. And then most DLP systems are not optimized for bioprinting. It needs large vat or container. That means it requires large quantity of cells and polymers. Next in this, using the same DLP, they can print complex tissue architecture, such as alveoli, the unit of the lung. Researchers have demonstrated that the oxygenated blood, when passing through the 3D printed lung air sac, get oxygenated as seen in this video. This is what the process looks like. That shows the deoxygenated blood pass through the vessels, air sac vessels, and air sac here mimics and works as inhalation, and exhalation happens to achieve oxygenation. However, these are prepared using without using cells. This is using PDMS-based polymer. So for the not for the implantation purpose, but for the analytical purpose. And this video here shows the complex lung mimetic design, where blood vessels are precisely designed to be surrounded by the alveoli sac. The entire system is with a hydrogel system and demonstrate native lung alveoli function. As we see here, we have both artery and venous, and that is surrounded by the surrounding the alveoli sac. Deoxygenated blood goes through one side, and then upon inhalation, exhalation, mimicking, it gets oxygenated. Next example in 3D bioprinting is inkjet bioprinter, which is similar to office printer. An actuator present in the printer eject the bio ink droplets containing cells as seen in the video. The limitation here in this one, the nozzle is so small, so only one cell can be ejected at the time. That means it can be easily clogged, and also printing bigger structure like organ or tissue is difficult with this inkjet bioprinter. Then we have laser assisted bioprinting. How this works is essentially similar to laser printer. Layer of hydrogen loaded cells start to suit as designed, and the exposed spot will be costly. The limitation of this method is we can have only one layer at a time. And also metallic sheet used at the bottom can cause easy contamination. Another recent example, and the most exciting one, is based on extrusion printing. The extrusion printing that we from the method that I showed earlier. This exciting work is published in Science, one of the highly reputed journal in the research field. It is called FRESH, Freeform Reversible Embedding of Suspended Hydrogels. Basically, they used conventional extrusion printer, nothing much different, but they used a solution of concentrated gelatin particle solution. So this solution has a lot of nanoparticles made of gelatin. And then the extrusion, they extruded collagen containing cells into this gelatin concentrated solution, so they can print literally any complex structure as seen in this video. Upon completing of this 3D printing, we can remove gelatin because gelatin is easily dissolvable, and you can get the 3D printed structure intact in a standalone manner. The same method first has been used to 3D print human heart. What we see here, a pediatric patient specific MRI data set of human heart, and this is the first based printed heart. As we seen here, the cardiac chambers, valves, and vessels are precisely printed using this FRESH technique. What we see here is a printed heart without cells. It's just the fabrication of heart. Like as you see, there is a inner chamber, and then inner wall, outer wall, the chamber, and then these wall has a lot of pores that can be loaded with cells. So in this case, they loaded cardiomyocytes, cardiac cells, into the wall, and the heart cells started to beat upon stimulation from an electric source. And then upon increase in the electric source, to the beat is increased as well. Most importantly, heart cells. So these results collectively inform us that the advancement is real, and the field bioprinting from growth factors, the biomaterials, and the delivery system and 3D printing, we are moving forward to create such as complex organs in the field. However, advancement in 3D printing technology is really high, but we are not able to print human organs yet, the real functional human organs yet, because multiple reasons. First of all, it requires billions and billions of cells to fabricate tissue, which is hard to scale. And then upon 3D printing, all those cells need to be alive. So we should preserve the cell's viability, which is again challenging. And then one particular cell is not purely available in one organ. We have multiple various types of cells are in the organs, and including all of them in the 3D bioprinting is extremely challenging. In addition, thus far, long-term effect is unknown, and also ethical concerns are there. Anyhow, comparatively less functional organ such as ear, cartilage tissue, or bone has been printed successfully. However, complex organs like lungs, kidney, and then heart will take a long time, around 15 to 10 to 15 years. And then finally, today in this webinar, we will discuss about the fabrication of organs on a chip system for drug screening application. So this is very, very important for pharmaceutical scientists like us. So drug development and screening is fully dependent on in vitro and in vivo model system. Before moving forward to clinical translation studies, in vitro models are commonly used. They can incorporate various types of cells, also biophysical properties, ECM features, and biochemical stimuli. But this system lacks of a more sophisticated whole organ culture system. On the other hand, we have in vivo models like preclinical models using animals. They can recapitulate the human organs as well as their response to investigational therapies. Restricted by the access to the test subjects and the feasibility of probing methodologies as well as the ethical concerns, they might not be accurate models of human issues. So microfluidic is the way to go to address these issues exist with in vitro and in vivo models. So what is microfluidics? Microfluidics is the science and technology of systems that process or manipulate small amount of fluids using channels with dimensions of tens to hundreds of micrometers. The very first application of microfluidics was in analysis. In this case, small samples required. The reagent quantity is also very, very small. In higher resolution, and it's highly sensitive, and it's low cost, short analysis time with a small footprints. So advantage of microfluidics over in vitro and in vivo models are biomimetic. They are biomimetic. Preservation of cell phenotype, heterotypic interactions can be achieved, increased human relevance, no ethical concerns, easy of handling, integration of detection and imaging system, and also low cost. And then microfluidics can be applied to multiple fields, such as lab on a chip, for drug administration, and also for cell analysis. And organs on a chip are microfluidic devices for culturing living cells in continuously perfused micrometer-sized chambers in order to model physiological functions of tissues and organs. The following video demonstrates the lung on a chip fabrication, and it's explained in the following video. Let's listen.
Ramesh, it is not audible. No,
Sorry,
it
is
is
it
not audible.
Aha. Seems.
We're just able to hear some small kind of beep sounds. Is that
Oh,
the
I see.
original? Yeah.
No, I can hear in my earphone, but okay. Probably something is wrong. I couldn't figure out what is the problem. Okay, if that's the case, how about let me move on to the next slide. And then I have another video show here.
Fine, fine.
Can you hear?
No, it's not audible.
It's not audible as well. Okay. Really sorry about that. think I can explain as the video goes on. So this is the microphysiological system, right? Let's okay. So human on a chip using with the knowledge gained from microfluidic device and organ on a chip, we can literally create human on a chip that have potential to model diverse diseases and ultimately replacing animal models for systemic substance testing. So what this video shows here basically creating small channels that represent individual part of our body, individual organs of our body. For example, intestine, liver, heart, and then also kidney. Those cells will be cultured in a specialized specified chamber. They are connected using a micro channel, and then they can be cultured, and then the drug can be tested using this system. So that is basically, I can share the link for this video. These videos are available on YouTube. They're really very educational. So I will share the link and you can share with the students. And then collectively, this is probably the last slide, I think. So together, pharmaceutical science and tissue engineering depends and benefits each other. And there is a vast opportunity to be explored in this currently advancing field. So as a pharmaceutical scientist, since we study much about biomaterials, drugs, pharmacology, we can pave the way to move forward, to develop, to engage ourselves in the research, to move this field forward. So tissue engineering and regenerative medicine is like ocean. It can absorb a lot of field like molecular biology, material scientists, pharmaceutical scientists, bioengineers. They all work together as a team to create functional tissue. So there's a huge opportunity for the students to move forward in that direction. So that's the end of the slide. And I would like to thank once again Arul sir, which is and the faculty and the organizing committee for having me here today. Thank you so much.
So participants, you can be free to ask questions or queries. Either you can post it in the chat or else you can directly, I mean, it's muted. So please post it in the chat if there are any queries.
One of my friend asked, commented about, please talk about the relationship to pharma research. So from this webinar, what I would like to say that when it comes to tissue regeneration, we use a lot of proteins or therapeutics. And then when it comes to proteins or any kind of therapeutics, the major important part is delivery. So how do we achieve delivery? That is basically, it's our job, right, to understand and to study about it, like pharmacokinetics and then pharmacodynamics involved there. So delivery system and then pharmacokinetics and then pharmacodynamics. That is one huge part, like, when it comes to growth factor delivery. It's a very huge market out there, growth factor delivery. It's not explored. One of the product approved by FDA is BMP2 for the regeneration of bone, and there are so many products are in pipeline to enhance to facilitate the regeneration of bone tissue. So where we can place ourselves in that direction. And then when it comes to microfluidics, the future will be microfluidics, because the Moderna based vaccine, they don't use virus particle. It's basically protein, the spike protein. They just develop spike protein, unlike Covaxin or Covishield, where we use live particle, inactivated live particle, right? But Moderna or Pfizer, they move forward to the next technology, which is mRNA particle delivery. They just deliver only the particle, cells recognize, and then they can fight against COVID. So that's how. So when it comes to microfluidics, so many researchers in the States during the COVID period, they used microfluidic channel to test the efficacy of the drugs, which bypasses the standard in vitro culture and also the preclinical model. We use lots of animals, right? A microfluidic can fulfill the need for the drug screening and testing. So students are advised to think about or study more about how microfluidics channels can be used for drug screening application. And then when it comes to bioprinting, which is very important in my opinion, because the standard drug delivery formulation techniques like capsule encapsulation or tablet, if you look, we have a different type of system to develop to prepare them. Now, in our case, we use DLP, digital light processing based 3D printing, that can print microgels in a minute in any shape we want. We can control the degradability, and then we can achieve number of production in a short period of time. So opportunities out there to identify the right technology to be included for the formulation. So see, formulation wise, pharmacology, formulation, pharmacology, and also drug screening. We have potential out there.
We have a couple of questions in the chat box. So from the first one, I think it's from Kogila. Covaxin have a clinical trials or not?
The clinical trial, I don't know exactly whether Covaxin had or not. From what I understand here, both Moderna is an American-based company, and then Pfizer, they have headquarters here, and they had clinical trial for number of patients. I do not have exactly the number, but in case of Covaxin, definitely without clinical trial, they wouldn't come to the they wouldn't come to the market. So definitely they must have had clinical trials. But now we are also in part of the clinical trial, right? For an example, what we do, like it will be in the multiple stages. So in the first stage, we cannot test the material in 100,000 patient, right? It's like we will be identify particular test group, and then we will test this material, test this therapeutic, and then move on forward.
Yeah, so the next one is from Anjali Sudhi. The question goes like this: how does advanced tissue regeneration work in pediatry field nowadays?
Yeah,
can any hope with
yeah, that's very true. One of the successfully applied patients are cleft palate patients. So they are like highly benefited, and a lot of dental related problems, lab made tooth can be implanted. And then when it comes to bone is the most successful regeneration thus far, because that many research have been done. So pediatric patients are not like from the perspective of tissue engineering. However, when it comes to organ transplantation or organ repair, mostly involve elder patients or patients who are vulnerable, who work in who are like soldiers. They are vulnerable, right? So number of bone injury happens more in the combat field. So we have patients like that. At the same time, every hospital, they have a pediatric reconstructive and regenerative pediatric department where they selectively study the use of tissue engineering application.
Okay, so the next question is very kind of very okay. Can this technique be used to increase cellular structure and create humans with superior strength?
Definitely, the way we are moving forward, it is absolutely possible, but not in the near future. It will be like far beyond. But we were thinking of, we cannot even think about create an organ 20 years back, but now we can literally create bone in the lab, and then we can literally create heart-like material using 3D bioprinting. So definitely possible, in my opinion, as a scientist who believes in science. All these achievements are with advancement in the field, they are realizable in the future. Definitely not in the near future, but definitely in the far future.
He's third year student of a college who has put the question.
That's
Right. Another one question which has come to me directly is, what motivated to research? I think it must be, what is the reason that maybe you would have concentrated on this particular field? I think that is what I've guessed.
So in my case, it's a different story, because I was more like a warder. I just followed the path, but researchers, like, they plan where they want to be. For an example, when I was working in GSK as a medical representative, my junior, I really have to thank him here. My junior, his name is Murugan, who invited me to Korea. So he was the one showed me the research path. Actually, he brought me to the state to South Korea. I was doing my master's, and I had no clue about research back then. So I just grasped the opportunity. Okay, I'm going for master's. But when I entered master's degree, the world out there about tiny particles, nano-sized particles, and we can create them in the lab. That's what actually motivated me. If we can create something like that, these can be applied in our field, which is pharmaceutical science. So I wanted to study more about delivery. So when it came to delivery, in my PhD, I was given a task about tissue regeneration, that is bone tissue regeneration. So I started utilizing this nano-sized material for the delivery of proteins for the regeneration of bone tissue. That's how my path came to the tissue engineering field. So, but for the students, I would say, don't follow anything. Whenever you have a chance, find out what is basically or currently happening. For an example, CRISPR will be the future. Any kind of cancer or like genetically hard to cure genetic disorders, people like scientists have been like going super hard with the technology of CRISPR. And CRISPR, basically, it's like an engineering system you deliver using particle. Again, it will be us, they use lipid nanoparticles with a different functionality. So these lipid nanoparticles can be injected. They can be taken up by the diseased cells or genetically modified cells. They will start correcting them, so the future will be there. So I would suggest our students to study more about CRISPR technology for hard to treat diseases. That would be one way. And then 3D printing is like fast moving field. So what we've discussed, extrusion printing came like 7, 8, 10 years before. But now, back then we could not fabricate anything complicated structure. But last year, this guy who published in Science, who created art, so the field will be advancing very fast. So if you search this journals called Science and Nature, you can simply browse what kind of articles they are publishing, and then you can choose your research path based on those articles published in Nature and Science or that kind of highly reputed journal. That will give you an idea of, okay, if I want to pursue research, what field I want to choose, or what direction I want to go. That will help you to decide.
I think truly motivated, really motivating answer, of course.
Vijesh?
Thank you.
Yeah.
Vijesh.
Yes, sir. Please.
Yeah, I have a question to Mr. Ramesh.
Sure, sir. Please.
Sir, sir, because you were there, I had to say this. I felt like I'm a student, so it's a beautiful, it's an honor to be here, again, once again, I'm really thank you for having me. Yes, sir, please go ahead.
Yeah. Ramesh, there must be some limitation of tissue engineering, right? Like a neovascularization or something.
Yeah.
How do you correlate neovascularization with the tissue engineering?
Yes, sir. So
how to overcome that?
Yeah. So when we say neovascularization, that means new blood vessel formation. That has been a tremendous problem in the field, in the tissue engineering field, because earlier people were using bulk gel, like they make like a bulk material, they implant at the defect site. Because of the material is bulk, it prevented blood vessel to invade into the core of the defect. That means no nutrient supply, no blood supply, the tissue ultimately died. That's why vascularization is very important. So in order to achieve vascularization, such as neovascularization, we need to provide some cues and also some spaces. So when we use porous structure, blood vessels can invade into the defect in the scaffold. And then we can also use chemical growth, biochemical cues like growth factor. So in our case, we used VEGF, which is called vascular endothelial growth factor, and then PDGF, platelet derived growth factors. These two growth factors basically recruit blood vessel forming cells to the defect site to form the neovascular structure. So blood vessel, creating blood vessels before the tissue regeneration happens is very important. Did I answer the question, sir?
Yeah, I got the answer.
Okay, yeah,
And
thank you.
you can also advise to our own students. Any advice from you?
I would really say one thing, sir, like us we, like when in India, when we were as a undergrad, we study hard but mostly theory, and those theory has been created, those principle theory fundamentals are like been fixed set like 20 years before, whatever have been found in the book form. But the current advancement, like whatever I talk about, 3D bioprinting, microfluidics, advanced delivery systems, they are in the research setup, right? They are not in the book. It will take for years to be on the book of our own syllabus. So I highly recommend our students to read Nature, or there are a lot of pharmaceutical journals also there, Nature, Science, and then molecular pharmaceutics in ACS, and then European Journal of Pharmaceutical Science, I think. So they have a lot of pharmaceutical science journal as well. Go read at least one paper in a week. That will be my advice to our students. Even if you are interested in research or not, explore what is happening in the research world, considering our field. So in the pharmaceutical industry, is anyone finding something new or any drugs being invented? All those things we need to go and search for them. That will be my advice.
Thank you, Ramesh, for being with us. And over to Mr.
Yes.
Vijay, the coordinator.
Yes,
Yeah,
thank
yeah,
you,
sure.
sir. Thank you,
Thank you,
sir.
sir.
And so actually what Ramesh sir says is the students has to develop the habit of reading with research attire, right? So
What is that, sir? Can you go ahead again?
what I was saying is students has to develop the habit of reading
Yeah,
with the research attire. No,
yeah, very true.
that's right. So, shall I go to the next question, sir?
Oh yeah, sure, please.
So this is from Nabila. She's asking, what is the latest advancement of tissue engineering in osteoblastoma?
Yeah, so this is again a very interesting question. I had some slides to discuss about cancer related one. So currently in our lab, we are developing this lab made bone material, and then to understand how cancer formation happens within the bone, which is really critical, because any cancer ultimately metastasizes into bone, right? How or what attracts cancer cells to the bone? This is like an unanswered question. So we developed a material which is called mineralized bone-like material in the lab, which we published in Nature Communication last year. We are currently utilizing that material to understand more about cancer invasion, and then like osteoblastoma. To understand is one thing, and then to develop treatment strategies another thing. So in this case, we are in the understanding situation. No, I wouldn't say, we are in the stage of understanding osteoblastoma. So in the future, if we know how we can cure, tissue engineering can be a tool. For an example, if there is a cancer tissue, we can excise them and then we can implant a newly formed bone tissue. That is one way to go. But for the cure itself, we may have to find something else, which is different from tissue engineering field.
Okay. All right, sir. So the next question is from Rasmi Sharon. It's already been answered, but still I'll just ask it here. So she asked, how far is medical science from generating a whole human body by tissue engineering? Is it possible to think about it?
Yeah, I think as we discussed before, there is a scientist in China who cloned human, right? So, and it was, how do we say it? He successfully created human in the lab, which is ultimately possible. That is, cloning is a way to go, and then tissue engineering is a different way to go and pursue. So we are, as I said somewhere in the slides, it's a budding technology. So it takes close to 50 years probably to create like functional organ that can be put together to create a human being. That it's like it sounds like a sci-fi movie, but if we can create organs which is functional in the lab, then putting them together will be possible, which will be feasible in my opinion. But as I said, it will take probably a century or like who knows.
All right. Okay. Fine, sir. And now another interesting question. What are the major demerits of Indian education system with other, especially in pharma field?
Yeah, this is a the problem. That's because we never increase our expectation, or like we never fought for it. I think, for an example, I think, I don't know the exact number, but if you look at the market value, most of the therapeutics produced or supplied in the world are produced in India. So it's a huge market. But pharmacists are working in manufacturing, even if they are working, how much they are paid. So it's like highly underrated field. That's why, in India, the way in a way, if I put it, the pharma field is not, we are not teaching our kids to handle or to manage the pharma field. That's what I feel like. We are educated, we know about drugs and stuff, but are we mature enough to manage like pharma? It's a big field, right? Like pharma companies, or like what will be the role? Will we be the deciding authority? That is missing in our, in our education system. So we need to include something about management in addition to educating about pharma. We need to teach them about management, to take this to the next level. And then in other terms, demerits, I don't think there is any demerits. We are equally knowledgeable as others from other countries. So I wouldn't say there is demerits in pharma education system. We definitely need to include current advancement that is happening in pharma. That's probably one weakness that we have. If we include that in addition to management, collectively together, it can push the pharma field further in India.
Yes, hope that in the coming years we get that change of the real research.
Yeah.
Thanks a lot.
Then I have another. Nabila also asked about latest advancement in osteolysis, okay. Which is again that is again a very important question, because osteolysis that directly causes osteoporosis, which is a very big concern. Meaning, body recognize our own. It's basically bone remodeling, we call it, right? As the bone formed, macrophages comes, attacks, they degrade bone, and then the new bone forms. So if there is a miscommunication during the bone remodeling, leads to osteolysis, that is osteoporosis, and then breakage of bone. So we need to understand why macrophage does that, and then why our body do not settling or do not initiate bone formation as soon as the resorption happens. So in order to achieve that, we need to find there is a medicine bisphosphonate. So bisphosphonate is basically used for osteoporosis to prevent osteolysis. Exactly this case. What that does, it prevents macrophage macrophage resorption of the bone. That is exactly what it does. Now look at this way. If we have a macrophage lysing the bone, that then our body should initiate regenerate the bone. So we need to give some chemical stimuli to initiate the bone formation. So osteolysis, there are some treatments available, and then identifying even much more capable therapeutics will be an ideal cure for preventing or like treating osteolysis.
Okay, thank you. So
yeah, this organ on a chip technology, Shyam Krishna asked about, can organ on a chip technology used to treat neurological disorders of brain? So organ on a chip, is the concept of organ on a chip is basically to identify or to understand how things are happening in the body. So to understand that, we need to have a real animal to understand, right? But with organ on a chip, we can create a small organ-like structure in the chip, and then we can test or we can screen the drug. So that will be a tool to develop drug. For drug screening, organ on a chip will be used, but they will not be directly used for the neurological disorder. But implantation using microfluidic channel or microfabricated devices can be used for the neurological disorder. Slightly different. Yeah. Yeah. So Satya is asking about some journals representing real advancement of pharmacy and also in the department of pharmacognosy. There is this journal called Molecular Pharmaceutics. It's one of the journal in American Chemical Society publication group. They publish a lot of pharma-related articles. The highly advanced drug-related one, we have something called drug discovery today. You can subscribe to their notification alerts, email alerts for every issue. They will send you the list of articles published. So drug discovery today, I would suggest, and then molecular pharmaceutics is another one. And then European Journal of Pharmaceutics, International Journal of Pharmaceutics, they are also will be useful as well.
So I think that was the last question.
Okay.
And,
yeah.
And I He's gone.
Yeah. No, I hope I addressed. Some questions are like even if I don't address, even if I have not addressed properly, please reach out to me later through email. We'll be happy to help. Happy to address those questions.
Yes. All right. Thanks a lot. Thanks a lot, Ramesh sir, for that query session as well as your presentation. Really added value. It made our day. It made our day.
Thank you so much. Thank you.
Okay.
Really.
Thanks a lot. Thanks a lot.
Yeah.
Ramesh, we are all enjoyed the day.
Thank you, sir. Yeah, let's do this quite often, sir. Like we can come up with different topics, and then
we'll call you.
If we can,
we'll disturb you.
Yeah, we will. Yeah,
we'll
sure,
sir. We'll keep
happy disturb. Yeah, I'm really proud that like I didn't expect this many questions would come from our students. So some of the questions are really like to the point. So I'm really happy.
Yeah. Thank you.
Thank you, Ramesh. Regards to your
Thank you,
wife.
sir. Thank you so much. Yeah. Sure, sir. Kandipa solren, sir.
Okay.
Okay. Right. Yeah. Bye.